Good afternoon. My name is Tess Taylor, and I'm a specialist in the Community Education Department at MDA. We're thrilled to have you join us today for the first day of our MDA virtual learning series on CMT, Charcot-Marie-Tooth disease. This learning event is part of our larger MDA community education program, which focuses on bringing the neuromuscular disease community together around education and resources. Be sure to visit the community education section on mda.org for updates on upcoming events. Just so you know, we are recording today's event and we'll be posting it to the website for on-demand viewing at a later date. Everyone registered for today's session will receive that information in their inbox. Please know that all phone lines have been muted, but you can still ask questions. We will have a Q&A session at the end of each presentation. However, they will be a little bit shorter today, but please still submit your questions in the chat, and we will make sure to address as many as possible. For nearly 75 years, MDA has led the way in funding research to accelerate the development of new treatments, expanding access to care and clinical trials through the MDA Care Center Network, and advocating for access and inclusion for the neuromuscular disease community. MDA delivers its mission through research, access to care, advocacy, education, recreation, and engagement. Since its inception, MDA has invested more than $1.1 billion in neuromuscular disease research. Our MDA care centers are located at over 150 top medical institutions nationwide, providing care from clinicians who specialize in neuromuscular disease. MDA's advocacy work focuses on support for accelerated approvals and novel research, access to healthcare, accessible air travel, and more. This focus plays a vital role in shaping laws and policies that support the needs of people living with rare neuromuscular diseases. Our community education and recreation programs are designed to connect individuals and families. We're committed to building a nurturing environment where our members can share experiences, triumphs, and challenges. Every year, we offer over 40 educational programs, over 20 summer camps, and dozens of other community activities, both in person and virtually, like our one today. If you haven't done so already, we invite you to join the MDA community by registering with us. Registration is free. Please see the link in the chat. And to learn more about MDA, please visit mda.org. So this is the first day of our two-day event regarding CMT. We have three presentations planned for today, beginning with Brian Lynn, PhD, the Research Portfolio Director at MDA, and Allison Moore, who is the CEO and founder of HNF, who will be briefly discussing the current efforts in research and care for CMT. Following that, at 4 o'clock, Dr. Ryan Kostora will present on medical and symptom management for CMT. And our last presentation today will be Shauna Feely at 4:40. She's a licensed genetic counselor and will be here to talk about genetic testing with us. So MDA is proud to partner with the Hereditary Neuropathy Foundation, HNF, for this special two-day event. And without further ado, we would like to take some time to discuss HNF and MDA's current perspectives on CMT. So I'm pleased to introduce Brian Lynn, who is MDA's Research Portfolio Director. Thank you for sharing some insights with us today, Dr. Lynn. And Allison will also be hopping on to provide us with some of HNF's perspective on CMT. Allison, again, is the CEO and founder of HNF. So, Brian, I will go ahead and turn the time over to you and Allison, and thank you so much.
Yeah, thanks, Tess. And so, hi, everyone. It's a pleasure to be here today, especially with the Hereditary Neuropathy Foundation, to discuss some of the CMT advances. And so let me share my screen. So this is the agenda for the next 20 minutes. I'm going to start with a brief overview of MDA's role in CMT research, and then I'll speak a little bit about, where are we now in terms of getting therapies to the finish line for CMT. And then I'll round out this talk with hitting on some of our MDA research highlights, some of our MDA funded work. And it will fall on a few key areas: genetics, some of the new therapies that are on the horizon that we should really look forward to, and our efforts in clinical trial readiness. And so Tess had a great overview about the MDA, but for those who aren't familiar with our history and our history with CMT, we're an organization that was really founded in 1950 by a group of families with muscle diseases. And our portfolio really over the past many decades has expanded to over 330 different neuromuscular diseases, including CMT. And we started funding research, I think, in CMT, really accelerating discoveries in 1970 and then into the 1980s and so on. And one of the major breakthroughs that we funded really occurred in the late 1980s. And you can see this on the right here, where MDA-funded researchers were actually the first to identify the first CMT gene for CMT, and that was for CMT1A. And so you can see some of these seminal studies that came out. And so the one on the left was published in 1989 by Dr. Vance and his colleagues at Duke, and they linked CMT1A to the human chromosome number 17. And then a couple years later, another group of MDA-funded researchers, Dr. Lepsky at the Human Genome Center and his colleagues, were then able to discover that it was actually a DNA duplication that was happening on a gene called PMP22 that actually causes CMT1A. And it was really from these discoveries that really, kickstarted CMT research and accelerated the field. And over the last 20 years, we have invested over 30 million into CMT research to kind of drive CMT discoveries. And the goal of our MDA research program is to do everything that we can to accelerate therapeutics developments. And we're in a really exciting time for neuromuscular diseases, where we now have 28 approved drugs, and 10 of them were actually directly supported through MDA research. And you can see them listed here. And we're hoping that CMT will join this list fairly soon. And so just a brief overview about CMT. We know that CMT is the most common inherited neuromuscular disease. It affects about 1 in every 2,500 individuals. There's 130,000 people in the US who are living with CMT, and it's a peripheral neuropathy. And so it affects the peripheral nerve that you can see on the right here. And the purpose of the peripheral nerve is really to send signals out to the muscles, to innervate them, to tell them to contract, and so on. And kind of two key parts that I want to key in on are the main part, or the extension of the peripheral nerve, is called the axon, which is this long projection. And this axon is actually wrapped by what's called a myelin sheath. And so if you think of the peripheral nerve as kind of like an electrical wire, and the axon is the projection of that wire, and the myelin sheath is kind of like the insulation around the wire. And researchers have identified over the past many decades 169 different genetic subtypes for CMT. And these are often classified into numbers or letters based on their function or their inheritance or where they affect the nerve. And so, for example, CMT1 is often called demyelinating neuropathies because they directly affect the myelin sheath. And so you can see here, for example, the mutations in CMT1A cause demyelination or breakdown of this myelin. And CMT2 is called axonal because they affect or cause axonal damage. And then there are many other subtypes as well, such as CMT4, and these are often different, that they're more severe or they're rarer and so on. And so there's many other subtypes as well. And while we do know many of the genetic origins of many of the common types of CMTs and some of the rare forms, some of the challenges of getting therapeutics across the finish line are kind of intrinsic to the disease itself. And so we know that symptoms and severity of the disease can really vary. They can even vary from person to person, and those with the same subtype of CMT, for example. And most cases of CMT are also slowly progressing, which makes it a little bit challenging for clinical trials, where within that kind of 2-year window that you have the trial, you need to be able to detect or a change due to that treatment. And so that requires very sensitive tools in order to measure the changes in disease progression. And as a result, currently we don't have any approved therapies, but we do know that multidisciplinary care for CMT is really critical. And so where are we kind of now in the development of treatments for CMT, and how did we actually get here? And so if we look a little bit at our history, in the early years, from the 1980s to the 2000s, much was really focused on gene discovery. So identifying genes associated with CMT, and we hit a lot of the major common genetic subtypes. And in the 2000s, a lot of it was dissecting how do these genetic mutations actually cause disease. And so those were studied in the preclinical stage, studying them in labs and animal models to really understand the underlying molecular mechanisms. Then in the 2010s, while most of the genetic research continued, there were more focused efforts towards building the foundation that we need to test those treatments. And so what that means is that we started creating patient registries and we started running natural history studies to track how CMT progresses and develop reliable outcome measures and markers of the disease so we can know whether the therapy is working. And then now in the 2020s, we're still building on that groundwork and we're still trying to ensure that the clinical trials are really set up for success. But we've also entered a phase of innovation where we have exciting new genetic technologies. There's gene therapies I'm sure many of you have heard about. People are exploring gene editing techniques and all these other innovative therapeutic strategies. And so we're really in this age of developing therapies that actually target the underlying genetic cause of the disease. And so all of this really brings us to the present day, where our MDA portfolio of research really includes an array of these various initiatives and studies, some that are focused on genetic discoveries, and then others that are focusing on bringing new labs and lab discoveries and treatments and bringing them to the clinic, and hopefully having the clinical trial readiness to support the testing of those treatments. And so I'm going to hit on some of those highlights of our MDA research portfolio. And so first, starting with genetics, we know there are now 135 CMT genes, and they span 169 subtypes. They're inherited in many different patterns. The majority of the ones that we know of are autosomal dominant, meaning that in order to have CMT, you would have to inherit one copy of the defective gene from one of your parents in order to have that CMT. But while more genes will be discovered over the next few years, there are additional challenges that we need to be able to tell whether a mutation that we identify newly or one that we know of in the past, whether it's actually disease causing or not. And so these are often called variants of uncertain significance, where we can identify these mutations through genetic testing, but we don't actually have the scientific evidence to say for sure, is this a part of the disease or is it benign and not part of the disease? And so we have been funding 2 studies recently to help kind of clarify these variants of uncertain significance. And one of those is founded by Dr. Stefan Zuckner at the University of Miami. And his platform is called the Genesis 2.0 project, where it aggregates a lot of patient genomic data and phenotype data, so clinical data. And it helps us basically, if we have a variant of interest, be able to see in all these patient genomes across the populations of CMT, how prevalent is this disease causing variant or this potential variant? And if it's very prevalent, it's more likely that it might be associated with disease rather than not. It also provides functional evidence integration, which means we can link this to lab studies. And so it's annotated with some of this data and annotated with patient outcomes. And that can help to support or refute whether that is a pathogenic variant or not. And then the second study that we're funding is to Dr. Charles Abrams. And he is an expert with CMT1X, and he's at the University of Illinois, Chicago. And so Dr. Abrams is working specifically to identify variants of uncertain significance in the gene GJB1 that causes CMT1X. And so he is developing a pipeline in the lab, functional assays to really look at each individual variant and say if it's functional or not, is it disruptive? And so he has been able to really clarify a bunch of these variants, and now we know whether they are pathogenic or they should be labeled as benign. Okay, and so next, it's also important to remember that we shouldn't just focus on, just the common subtypes of CMT. And so some of the work we fund is focused on some of the more ultra-rare CMT subtypes. And so 2 of these studies that I'm going to highlight, the first is Dr. Alessandra Bellino, and she works at the San Rafael Hospital, and she's specifically studying CMT4B, which is a rare recessive form of CMT that's typically early onsets, and I think we know about a few hundred cases across the world. But she is studying the cellular mechanisms that underlie CMT4B, and she's testing whether some of the existing drugs that we know of can help to restore some of that nerve function. And then Dr. Eric Storkenbaum, who's at the Radboud University, which I believe is in the Netherlands, he's studying a group of CMTs that share like a similar class of genes that encode a protein called tRNA synthetases. And he wants to see whether replenishing levels of the protein tRNAs are able to help with all of these various CMTs. And so he's studying a bunch of animal models and testing some of these therapeutics. Then a form of CMT that has made considerable progress over the last decade is for CMT2C. And so our funding over the past decade has really directly supported kind of a step-by-step effort to advance a therapeutic for this version of CMT. And this research effort is really led by Dr. Charlotte Sumner, who's at Johns Hopkins. And she was the one who actually discovered that CMT2C is caused by a genetic mutation in a gene called TRPV4 or TRPV4. And from her first grants with the MDA that started in 2013, she investigated in animal models how these mutations actually cause peripheral nerve degeneration and found out that it was because these TRPV4 proteins were actually overactive in the neuron and they were causing deficits. And then with her second grant that happened in 2019, she evaluated whether a TRPV4 small molecule inhibitor, so a drug that actually helps to suppress its overactivation, can help reduce some of the disease phenotypes. And so what she saw was that giving this TRPV4 inhibitor to mice was able to reduce some of their neurological deficits. It helped with what's called a leaky blood nerve barrier in the animal models. And so it really helped reduce some of the neurological aspects of the disease. And so Dr. Sumner is currently working with Actio Biosciences to advance a therapy, TRPV4 inhibitor for treating CMT2C. And they actually just dosed their first patient earlier this year in March in a phase 1 clinical trial. And then with her latest grant, it's really all about advancing clinical trial readiness for CMT2C. And so we funded her to help with developed her patient registry to define the progression and disease severity of CMT2C and develop outcome measures that can be effectively used for clinical trials, including studies on voice changes and MRI studies. And then one of the most exciting areas of therapeutic research for CMT right now is gene therapies, which I'm sure many of you have heard about. And so these treatments often use something called AAVs or adeno-associated viruses. And so you can think of AAV as a tiny delivery vehicle that carries instructions. And these instructions, when they're delivered into the body, can then tell the cells to produce a protein that is missing or suppress a gene that's working too hard and causing a bunch of issues. And so for CMT, you can see that there are many different gene therapies that are in production. They're in preclinical development for the majority of these. And so I'm going to highlight one in particular for CMT1X that we actually funded a few years back. And so we funded a grant to Dr. Cleopas Cleopa at the Cyprus Institute of Neurology and Genetics. And he is an expert in gene therapy and in CMT. And so we funded his studies to test his gene therapy in animal models of the disease. And so he tested his gene therapy in 2 mouse models that express a mutation in the GJB1 gene that causes CMT1X. And what you'll see is that in this mouse with the mutations, they have what's called abnormal myelination or demyelination. And so you can see this clearly from this microscope image of the peripheral nerve cross-section, which is basically if you think of the nerve as a string and then you cut it and then you look at the inside of it, this is what you would see. And so the circles are kind of like the axons, the actual nerves, and then the blue rings are the myelin. And you can see that there's a variety of different thicknesses to the myelin. It looks a little abnormal. And then when Dr. Cleopa gave the gene therapy to the mice, you can see it caused re-expression of that particular genetic protein, and it caused the myelin to look a lot healthier and rich. And this also improved a lot of the motor nerve conduction velocities. It improved muscle strength. And so this was really an encouraging sign that there may be a potential therapy on the horizon for CMT1X. Now, there are some challenges that still remain for gene therapies and to moving them to clinical translation. And so some of the outstanding questions are kind of, will these changes that are happening in the animal models translate over to humans who are much larger than rodents? And can it actually reach kind of the peripheral nerves? And then we want to make sure that these gene therapies are also safe for humans as well, and that it doesn't elicit any major immune reactions. And so those are some of the ongoing challenges. And so we are actively kind of discussing how to address these. And so we host an annual summit every year that we convene companies, collaborators, researchers to discuss some of these challenges. And at the conclusion, we publish these findings in a peer-reviewed journal. And so if you're interested, feel free to check those out. I think those are open access and you can read a little bit about gene therapies. And then also to address some of these challenges for gene therapies, we also funded fairly recently Dr. Kagiava, who works very closely with Dr. Cleopa at the Cypress Institutes. And so one award is for Dr. Kagiava to test whether she can use ultrasound to help these therapeutics actually reach the peripheral nerves easier. And then the second award is really to test whether she can develop a non-viral method. So the gene therapies were using adeno-associated viruses, but she wants to develop a non-viral method to actually deliver these gene therapies to humans. And this could potentially be safer, and it can also be redosed. Okay, so now moving away from therapeutics and towards the clinic. So across kind of CMT types, one common goal is really to ensure that we are clinical trial ready. And so we awarded a new research infrastructure grant to Dr. Joshua Burns at St. Jude's. And he and his colleagues previously established a suite of clinical outcome measures to evaluate the effectiveness of therapies. And so you have a variety of tests for pediatrics, infants, adults, and so on. And these are well validated. But one barrier that exists towards progressing towards kind of multi-center site trials is that we don't have enough kind of well-trained clinical evaluators in the space to actually collect these measurements. And so the goal of this grant is really to expand clinical evaluator training resources, especially to countries and sites, maybe not English-speaking sites as well. And so to have resources that are in different languages. And so when trials begin, we will have a certified and reliable network of clinical evaluators to really assess the treatment. And then additionally, a challenge for clinical trials is that, as I mentioned earlier, CMT progresses slowly in most forms, and that can make it really difficult to measure these changes over a short period, which is typically clinical trials are quite short, about 2 years or so. And so we funded Dr. Mary Riley at the University College of London Institute of Neurology and her research to really validate whether we can use MRI as a way to track disease changes. And so the idea here is that when muscles atrophy during disease progression, fat starts to infiltrate those spaces. And so one idea is to use MRI to measure that fat fraction or that fat infiltration as a measure of disease progression. And so from the study, Dr. Riley has effectively been able to validate using MRI to measure the foot in people with CMT1A over 12 months. And that's a sensitive way to monitor disease progression. And she's also been able to validate MRI measurements in the calf and thigh and fat infiltration in those and various other forms of CMT like CMT1X, 1B, and 2A. And you can see an example of this in this image here, where this is the healthy individual and the black areas are the muscle, and the top here is the thigh and the bottom is the calf, and the white is the fats. And so you can see in a healthy individual, maybe about 2% of fat is infiltrating, but in the individuals with CMT, especially in the calf, you can see there's a lot more white, meaning that there's a lot more fat infiltration in those muscles. And so this is one way to that could potentially enter clinical trials and be a way to measure treatment effects. And so if you want to see or read about more information on these MDA grants and on the MDA research, feel free to check out our webpage at mda.org/science/grants@a-glance, and you can click on each of these individual research projects and learn a little bit more about them. I also want to just briefly highlight that we support a lot of scientific and patient meetings throughout the year to help really connect researchers and patients and the community together. We're going to support a researcher meeting on CMT1E that's happening later this year, where they're going to talk about how to develop treatments for CMT1E. And I also want to highlight that our MDA clinical conference, our professional conference, is happening next year from March 8th to 11th. And we're going to have a 2-hour session on inherited peripheral neuropathy and new therapies. And so please check out more information about MJ conference at this website. And so just want to say thank you, everyone, for listening, and thank you for your support. Really appreciate it.
Thank you, Dr. Lynn. And now we will have Allison Moore, who's the CEO and founder of HNF, and she will give her perspective and HNF's perspective. Thank you so much. Hi, Allison. I think you're muted.
That's funny.
Happens to all of us.
I guess I have to repeat. I want to thank Brian and the entire MDA for inviting me to present today. It means a lot to me and the HNF. I also want to do a huge shout out for the $30 million that have gone to CMT research. As a mother, a patient, caregiver, an aunt, a sister, and so on. It's really important that we come up with treatments for CMT, and I think we're getting very close. And just to add that for today's talk, we're primarily focused on clinical trial readiness. So again, thank you for having me. So CMT won't wait, neither should we. So we're aligned. We're trying our best to get treatments into patients. Why is this not moving? There we go. So for those of you who don't know me, I'm the founder of HNF. I was diagnosed with cancer back in 1995. I was in my 20s, and I was given chemotherapy, and I had severe onset of CMT1A. Literally, it was 25 years of progression in 6 hours. So we started a program called CMT Deploy back in December. And it's an ambitious initiative to accelerate the development of CMT treatments and de-risk clinical trials. There are many active trials for those that don't know that, but they're still very high risk. We're still lacking in the area of being clinical trial ready, and the data and systems aren't completely in place. We do hope that most of these will get over the finish line, but to de-risk them, we're moving into another paradigm to modernize our natural histories and many things that we're doing. So this is something I think we all can be very excited about. As being diagnosed with CMT for so many years, we've never seen so many pharma companies in the space. So it's our job, not only like what MDA is doing to get this basic science and translate therapies and get pharma interested, both small biotech and big pharma, but also we have to support them to make sure that these trials don't fail. So how do we do that? Obviously, there's a problem. There's 160 subtypes. I know, Brian, you said 135, but maybe your slides were a few weeks ago. There's new genes being founded all the time, and we included the ones that are not just Charcot-Marie-Tooth disease. One could be hereditary neuropathy, peripheral palsy, or some of the names that just are not called Charcot-Marie-Tooth. So this is inherited neuropathies. Clinical trials potentially can fail if the data is not clean, if it's not collected consistently. And what's very, very important is we make sure that data is regulatory ready, and we're aligned with the FDA and some of the international regulators in the world. As we all know, CMT is a complex disease because there's many symptoms that we experience. And sometimes it's really hard to see change in a short period of time, even a long period of time, which leads to biomarkers. Biomarkers are very important. Biomarkers are a way to measure change. And right now, although there are some, we need more. So what is HNF doing? We're trying to rally the troops. We're trying to bring the siloed data together. We're trying to collect data in a more robust fashion to make sure that it's consistent and it's very clean. And we're looking to expand to many, many collaborations, connecting the dots. MDA and HNF have been working together for some time, and as we move on in the presentation, you'll see how we kind of worked off of each other's programs to advance to other stages. So this is a really nice bubble chart that kind of shows what CMT Deploy is about. In the center is CMT Deploy. There's 4 main parts to this. It's the research hub, patient support, clinical trial support, and FDA. And as you see, as we expand, we go into different areas, and every piece of this puzzle is important to ensuring success. Because we have so many patients on the line and families and moms and dads and some young people, we do have a lot of resources. So we have a pain guide, we have an AFO guide. I think you're going to hear from Mitch Warner tomorrow on things that you can do to impact quality of life, stay healthy while we're waiting for these medications and gene therapies. So once again, connecting the dots. We have to collect data. We have to take this information. We have to learn from it. We have to take those insights and we have to take wisdom out of this. We need to make impact. We have to make sure that we sync the patient data to the clinical data, and we need to educate and make sure that we're there to support the CMT ecosystem to make sure that these trials are successful. So back in 2013, HNF formed the Global Registry for Inherited Neuropathies, and we collect not only patient-reported outcomes, but we also collect clinical outcomes. And we measure gait and foot drop, upper limb movement, and recently we've gotten into incorporating digital devices. As we all know, sometimes CMT, you may have a really good day or a really bad day, and the clinic is not the only way to measure CMT change, whether you're getting worse or better. We need to look at how CMT impacts us at work, at school, at home. And that's very, very important. And with technology today, we can do that. And I noticed there's some people from Australia and other countries here. We are now in 4 languages. We're expanding to 2 more languages. Sky's the limit. So it really is the global registry. We are open to any country putting patients into the registry. To give you a little snapshot, this is little Jackson. I just want to say that Jackson has a very severe form of CMT called CMT6. Not only does it cause mobility issues, but it does cause intellectual disability as well as blindness. But we like to show that he's a guy that we're getting closer and closer. We're working on basic science, preclinical gene therapies, HDAC6 inhibitors to try to help this group of patients. So GRIN collects a lot of stuff, right? We collect demographics, we collect the clinical assessments, the digital health, we look at drop foot. So when you're in the clinic, we have a sensor that goes around the ankle. So we can look at drop foot when patients are doing what they call the 6-minute walk test, which is part of the CMT functional outcome measure. We're now looking at breathing because certain types of CMT like 4C, 1A, not all patients, but some have issues with breathing and voice and so forth. So we want to be sure that we're looking at this because that may be an outcome measure later on when we're looking at change in a patient. We've also done some work with video assessments. We like to say words are 1,000 words, or I forget the saying, but video is very, very powerful. When you're looking at video, you can really see change with your own eyes. And I think that can be super impactful as we show the FDA change in these patients by looking at them, like walking up the stairs, for example. Very hard to measure in a clinical setting. Patient-reported outcomes are also super important. It's not just how you're functioning, but it's how you're feeling. And we believe that the patient voice is one of the most important things to focus on because what may matter to a physician may not matter to a patient. So we do a lot in this area with patient-reported outcome measures, impact on quality of life, sleep, gait, falls is a big one. We all are very upset when we fall and we risk injury, so it's super important. So we have the infrastructure to collect all this information in a systematic way, and we share it. We share it with the right people so we can make sure that this data is meaningful to the people in the ecosystem that are either developing drugs or eventually will approve drugs. So we believe CMT Deploy is the solution, and it started with the registry. Collecting real-world data is super important. We also have expanded. It's been about 2 years. We have a biobank. I believe on at the last slide, there's a link. We offer anywhere from 10 to 12 roadshows a year where patients, if they live in the geographic area where we're collecting its urine, blood, and plasma. It doesn't cost anything. You would have to get to the site, but you'd have the opportunity to participate in research. And we ask that you join the registry because not only are we looking then at patient-reported outcomes and also functional outcomes, but we're looking at what they call surrogate biomarkers. And that's a very attractive piece to the puzzle when FDA is looking at drugs. If we can look at something in the blood that if you take an intervention, it would change. And I like to use cholesterol as an example, right? Our cholesterol gets really high within a couple weeks, when you take a cholesterol medication, you go back and you do your blood work, you instantly see or you hope most times, your cholesterol goes down. And it's a beautiful biomarker to show the impact of a drug or a gene therapy. CMT certified evaluators is part of our program. Thanks to the MDA for kickstarting this with Dr. Joshua Burns. We've been able to work closely with Dr. Burns and some others on his team to get more and more evaluators within these centers of excellence. And why is that important? Because when these clinical trials are going on, we have to make sure that the PTs and the OTs are consistently measuring these patients with these CMT scales in a unified way, and they know what they're doing, because if you digress, the data may not be clean. So the idea is clean data. So we de-risk clinical trials. We make sure, because a second or 10 seconds, 10-second improvement can matter in a clinical trial, and it matters to us as patients. My husband always says, if you could just have 5% improvement, how would you feel? And I said, fantastic. And not getting worse? Great. So that's really important. Back to CMT digital wearables, we're partnered with a company called Biosensics. We actually met them at the MDA conference 2 years ago. We became really close, the CEO and myself, and we decided to fund a pilot study. We went to MDA, said we met this wonderful group of people. Would you be interested in helping us? So at our summit on site, we conducted research, and we were able to take these wearables, not only the one I discussed about going around the ankle when you're looking at foot drop, but we sent patients home for 2 weeks with a pendant, which you can see on the far right, and a wrist where you see in the middle, for 2 weeks, where we looked at sway, we looked at gait, we looked at steps, we were able to capture falls of patients. It was really daily tracking, and it's a highly reliable resource. We were able to correlate it to other outcome measures. And now some of the CMT companies are looking at wearables in their trials. And the goal is to continue this research. In fact, at our second year summit, the first year we had, I think, 42 patients enroll. We had 78 at the last summit. And some of them were repeats. So that we call that longitudinal data. So we were able to see change, whether the patients fell more, didn't fall more steps, different things. So this is really, really exciting, and we're going to continue this study, and you'll hear about that in a moment. So this is what I was talking about with the training. We did it on site. Joshua Burns is over on the right. He's a wonderful expert in CMT. He's dedicated his entire career to not only training, but a lot of research in developing these outcome measures. So we were fortunate enough to take the work that the MDA did with Joshua. And with Joshua came up with Dr. Kayla Cordinet, who's one of HNF's fellows, to figure out a method to validate the best way to train clinicians. Is it virtually? Is it in person? And I believe they're looking at the data now, but it probably will be a combination of the two. So it'll be a way to make sure that we, as we're doing research in the ecosystem of CMT, that every site that's conducting research or clinical trials are CMT certified. So this is just showing you the CMT FOM is a composite score of a lot of things and this happens to be me, as you can see, where the 9-hole peg is one of the measures. And, many of us have hand issues. This is showing the 6-minute walk test, part of the CMT FOM. You can see the sensors around her ankle and her thigh. And this just gives you kind of a snapshot of what went down at this summit. So a shout out for the summit. We are hosting the next one. It will be our third clinical trial readiness summit in 2026, April 24th and 25th in Maryland. So if you're interested in attending and I haven't met you, we'd love to see you. And these are some support. This is a QR code for support. There's wonderful information there. I'm not taking credit for that. I want to do a shout out to Estella Lugo and Courtney Hollett. They're incredible. They've developed all these wonderful booklets, tools, and many things that are just should not be missed. It really makes an impact on us and our families, and especially our children. And that's it. Thank you so much.
Thank you, Allison. Wow, that was wonderful to hear HNF's perspective and a little bit more of your journey as well. A lot of the questions that we received were actually what role do patients play in moving research forward, and like how can small partners help? And I think you were able to answer a lot of that within your presentation of how exactly patients can get hands-on and really try to move the needle, so to speak.
Well, thank you for that.
So I think we will go ahead and take a few questions from the audience. We do have some time. And Brian, since you are still around as well, it'd be wonderful if you could hop in here. One of the first questions that we had was on a specific subtype of CMT. Is there any work being done that could impact those with CMT type 2O?
So CMT type 2O is caused by a gene called DYNC1H. I don't believe there is any major natural history studies or studies being done actively for it, but there may be some work in the preclinical stage that are being done in the lab where researchers are studying it. So I think that's something to stay tuned to.
I mean, I can also make a comment. So this is a perfect time and I don't know who said the question, but when you build the evidence that this is a rare subtype of CMT and you start joining these patient registries and you go to the roadshow or you can show up at our summit, we usually take blood and urine and plasma at our summit. It's an opportunity for you to get in these biorepositories. So, Brian and I could do a little research to see there may be some investigators out there working on it. And then I know when you have a certain variant of CMT, there's groups in Facebook and out in the social media sphere and engaging them to also do the same thing because we share our data with researchers. And if we see something, then, we can bring it to the attention of very smart people that are working in these subtypes. So don't give up hope because there are, as you saw, 4C is a very rare subtype and they're very close to the clinic. And that's because the groups came together and they just kept pushing. And maybe that will be the next MDA grant one day, or maybe we'll do some type of natural history, including CMT-O patients, more than just one. So good for you for asking and jumping in.
Yeah, wonderful. And thank you for that, because the second part to that question was, how do we help kickstart this work, right? How do we get this to the forefront? So I think you answered that just perfectly.
And Tess, I was just going to say one other thing, because I think it's important for people to know. There's a company out there called NMD Pharma. It was on our list. They're a CMT agnostic trial. Now, right now, they're only doing type 1 and type 2, but they may expand to other subtypes. So once again, you can email Brian and I can kind of tell Brian what we're doing for the subtypes that maybe you would be eligible at some point to participate in that trial if it makes it to the next stage. Sorry, Tess, I just thought.
Oh, no, that's why we're here, is to get as much information as possible. So thank you so much, Allison. We had a question come through the chat. What is the best idea for testing if you don't know your subtype?
Yeah, sure, I can take that. And so I guess genetic testing, you can go into the clinic and request genetic testing for CMTs. There's oftentimes neuromuscular gene panels that you can look at with the common subtypes. You can go over it with your genetic counselors and such to try to get a genetic diagnosis. Sometimes there will be you might come up with a variant of uncertain significance, and then that might require a little bit more scientific evidence to kind of clarify those. But that's one way to try to get a genetic diagnosis. And if it's one of the common subtypes, most likely it would show up.
Great, thank you.
I can also mention, I would imagine most people do have neurologists or physicians that are willing to test it. But as another option, we have something called CMT Genie. It's a patient-initiated program. So it's through a company called Informed DNA. Stella maybe can put it in the chat. It gives you the opportunity to meet with a genetic counselor. Stella, how much is it? Maybe you can put in there too. It's not astronomical. And they can order the test for you. And then they have a post appointment with you to go through the results, just as another option.
Great, thank you. I'm just looking at some of the questions that came into the chat here. So I have had genetic testing with no CMT type match. I am third generation of classic and progressive symptoms. Is there any way I can participate now? I believe participating in clinical trials.
You want to go first, and then
Yeah, I
I'll
can
give
go
my perspective.
first.
Yeah.
So a lot of, I guess, CMT trials right now are focused on individual subtypes, so you probably won't be eligible for those, but there are some that are, as Ali mentioned, would be disease agnostic or small molecule therapies that would be more generalizable towards the CMT population. And so you may want to talk to your neurologist or look at clinicaltrials.gov, for example, to look at clinical trials that you may be eligible. You can go onto that website and look at inclusion criteria and things like that to see if you might be eligible for some of those clinical trials that don't look at specific CMT genes.
You want me to make a comment too? So there's a really nice story with a company called Applied Therapeutics. There's a gene called CMT SWORD, and I think with Brian, with the funding that he had given to Stefan Zuckner at University of Miami, he has this database where they were looking at variants of unknown significance. And then through data, right? Data, they were able to find similarities. I mean, Brian, you know better than me. You may be able to share it in a more scientific fashion. And they were able to find the SOAR gene. So just getting into GRIN, getting into MDA's databases, we share data into the Genesis project. So the more data that's being looked at, so in the GRIN, we give you an opportunity not only to fill out the questionnaires, but you can upload your genetic report and then it's in the system. So we were all, as a community, all of us are very well aware about these variants of unknown significance. And I'll also say one other thing, there's a class of drugs called HDAC6 inhibitors. So it's looking not only at, it's mostly axonal, but they're looking at demyelinating too. But a lot of the variants of unknown significance, am I right, Brian, are axonal?
Mhm.
So
Yep.
in a perfect world, one day we may see that some of these drugs could be agnostics. You know, even if you're classic CMT clinical, there may be an opportunity down the road. And that's something, you know, Brian being an expert in the FDA with the, you know, regulatory and the work we're doing, it's something we do want to bring up to the regulators. It's called basket trials, where we start opening them up ultimately for all subtypes. But I don't want to pretend this is around the corner. There's a lot of work that has to get done. So we have to be realistic. But a starting point is getting your genetic reports into these databases, if possible.
Wonderful, thank you. And it also looked like Estella from HNF is put, and Marissa from MDA are putting some resources in the chat. So if you are live with us now, please get into that chat because there's a lot of rich discussion and resources happening there. So I did have another question that we were curious about. Would you say research in CMT is more focused on children or adults right now?
That's a tough question. I think it's focused on both. I think the majority start to be kind of, they start in late childhood or adolescence, but most therapies need to first be tested in adults before it can actually reach children. And so, in general, I guess in regards to therapeutic testing, it would be starting with adults, but in the future, we would want to test it in children because we think intervening at an earlier time in the disease course would have a better effect of that treatment in the end.
I'll also comment that for our digital health technologies, we started now bringing that into children. So Joshua Burns, he was at University of Sydney, now he's at St. Jude's. He has seen, probably followed over, I don't know how many decades, probably close to, I don't even want to say the number, but it's well into the hundreds and hundreds of patients. So they've been very much honed in on this. In the US, we're very well aware that we have to do more with children because I always say it's CMT starts in utero, right? We start having symptoms before we even realize we're having symptoms. So we've spoken to FDA about bringing these trials maybe into 16-year-olds. Maybe we can go even lower than that. But Brian's right. It's really is from a safety standpoint, they like to start in adults. But I also think we need to educate that CMT is a serious condition that deserves the attention. And if we don't treat early, then we may not get as much benefit. So as a community, we are well aware of this, and we're trying to bring a lot more data to show the importance of treating early. So to answer that question, there may not be interventions just yet for drugs, but there are natural history studies. So there's many opportunities for children to participate. And I think that if you have it in you to participate, your voice is heard, and more data gives us more ammunition when we're talking to the people that are in charge of these studies. I hope that makes sense, Brian.
Yeah, that's perfect.
Okay.
Great. So we had another question in the chat. What if your geneticist and your neurologist disagree on your subtype?
Whoa.
That's challenging. I would get a third opinion maybe from a genetic counselor just to look at your genetic test results and just get a third opinion and see what they say.
Great, thank you. Since we have a few more minutes, so I think we will take a few more questions. How long does it usually take research findings to turn into real treatments? And I know that's probably a pretty broad question.
Can you repeat that question again?
Yeah, so how long does it usually take for research findings to turn into real treatments?
Yeah, so that kind of varies a lot depending on some of the foundational research. And so really at the core of many of these genetic diseases, you really need to understand the genetic cause, understand the underlying molecular mechanisms associated with that disease. And so that can take a number of years. Usually this can take anywhere between 5 to 10 years in the academic setting. And then to take that finding and identify a drug could take another couple years. And then to validate that drug in preclinical models, such as animal models, and then to take it in clinic could take a few more years. And then through clinical trials, that can take even longer. And so, it can take a very long time. But I think with the technologies that we have today, with some of the hopefully regulatory aspects that are, a little bit sped up in terms of review and new platforms that hopefully are released by this new administration to help speed up the review of different drugs that use the same kind of technologies or those kind of things. This process towards drug development can be a little bit faster than it was in the past.
Great, thank you. A few more questions. Has there been drug repurposing screening done on any types of CMT?
Yeah, so there's a lot of researchers who are working on drug repurposing. I highlighted one, that was Dr. Alessandra Bellino, and she was working on CMT4B. And she was looking at a bunch of repurposed drugs that people have shown to be effective in other diseases or for nerve protection. And so, that research is definitely ongoing. And the benefit of that is most of them are approved. And so we know that it has a safe safety profile.
Wonderful, thanks. So we're running a little low on time, but we do have quite a few questions coming in, especially regarding like clinical studies. So if someone wanted to know if they qualified for a clinical study or they wanted to look into that, what avenues should folks take?
Yeah, I would talk to your neurologist. They usually have the detailed information about the clinical trials, the inclusion criteria. You can also look at clinicaltrials.gov and that would say exactly whether, you can look at the inclusion criteria that's listed there and see if you fall under those. But those would probably be the first steps if you want to look at clinical trials.
Wonderful, thank you. And MDA and HNF also put more resources in the chat regarding this, so the clinical finder tool, and then HNF you can also reach out to directly, as well as MDA's Resource Center. So I do think we'll wrap up your presentations here. I want to thank you, Dr. Lynn and Allison, so much for such great and rich discussion today, and for your perspectives on the state of CMT as far as MDA and HNF are concerned. So thank you all so much.
Thank you,
Thank you.
Tess. Thanks, everybody.